Hello, everyone. Today we'll be covering two chapters from our textbook, chapter 17 and 18, that discuss the two major components of our immune system, the innate and the adaptive immunity. We'll first begin with chapter 17, which deals with the innate immunity, also referred to as the nonspecific immunity. And immunity is the body's ability to ward off disease and protect against harmful environmental agents. It is what keeps us safe from infections and other threats to our health. There are two main types of immunity. The first is innate or nonspecific immunity. This includes generalized defenses that are always present and act against any pathogen, regardless of type. These defenses form our body's first line of protection. And as an example, our skin acts as a physical barrier, preventing pathogens from entering the body. If a pathogen does get inside, responses like fever help slow its growth and activate other immune defenses. The second type is adaptive or specific immunity. Unlike innate immunity, this system fine tunes its responses to a specific pathogen and can remember it for future encounters. This makes the response stronger and faster if the same pathogen attacks again. So, for instance, when T cells encounter a microbe, they can adapt to target that particular pathogen again. This targeted approach ensures a precise and effective immune response. Together, innate and adaptive immunity forms a powerful defense system that keeps us protected from infections and helps our body fight disease efficiently. The human body's immune system has three lines of defense to protect against disease. The first line of defense prevents pathogens from invading in the first place. These include physical barriers like the skin that we spoke about, but also mucous membranes, along with chemical defenses like stomach acid and enzymes and saliva, which help keep harmful microbes out. If pathogens get past this barrier, this is where the second line of defense provides general responses to fight infections. This includes inflammation, fever, white blood cells that attack invaders, so on and so forth. When a pathogen persists, the third line of defense activates specific immune responses. B cells produce antibodies, while T cells destroy infected cells and create immune memory for future protection. Together, these defenses keep the body safe from infection and disease. Chapter 17 deals namely with the first and second lines of defense, while Chapter 18 is concerned with the specific responses of the immune system. So, we'll begin our discussion with the innate immune system. The immune system has defenses that work non-specifically, meaning they act against many different types of pathogens instead of targeting just one specific type or one specific subtype even. Unlike adaptive immunity, these defenses have no memory. Each time a pathogen enters, the response is the same without long-term immunity. Many of these defenses are found in both vertebrate and invertebrate animals, meaning they have been present throughout evolution and are among the most ancient immune mechanisms. The body's first line of defenses are the initial barriers that protect against infection. These defenses, again, are the skin and mucous membranes and work to prevent pathogens from entering the body. Physical factors include barriers like that skin and mucous membrane, and they work to prevent pathogens from being able to get inside in the first place. An infection, if you think about the word, it really means that those pathogens are penetrating inside of the body. And so that could be through your skin, through a lesion, so on and so forth. We'll get into those details. But I just want to take a moment and just settle on this fact here. You have microbes that not only line the outside surfaces of your body, but what you think to be the inside surfaces, as we commonly talk about them in our day to day conversations. The inside of your mouth, the gastrointestinal lining of your body, even the respiratory tract. all of those that you think of as being inside your body are in fact outside of your body. The point at which your skin it turns inward towards your mouth and your sinuses and your ears, that still is considered outside of the body. Rather, when we penetrate those surfaces, your skin and your mucous membranes, that is technically inside of your body. And the microbiome that works inside of your gastrointestinal tract and within your respiratory system that don't cause infections are not actually invading your cells or invading your tissues. So, moving right along, we also have the chemical factors that involve substances like, again, enzymes, stomach acid, and antimicrobial proteins. We'll discuss more on those later. And those help to destroy, neutralize harmful microbes, and slow microbial growth. Lastly, as discussed earlier, normal microbiota do inhabit the outside surfaces of your body. And these are beneficial microbes. They live on your skin, they live in the gut, they help to prevent harmful pathogens from taking root and growing. Let's go ahead and go through the body's first line of defense and the physical factors that involved. Now, I already mentioned the skin and mucous membranes. There is also something called the lacrimal apparatus, and this is the apparatus that forms and drains tears from the eyes. And tears help wash away germs from the eyes and help remove and wash away debris and keep your eyes clean. In the respiratory system, there's something called the ciliary escalator. Its job is to move mucus and trapped microbes and debris upward, preventing them from reaching the lungs or being removed from the lungs. See, in the respiratory system, there are these cilia, or these tiny hair-like structures in the airways, and they beat at a certain rhythm and pulse that moves mucus and those trapped microbes and debris upward so they can then be coughed out or swallowed. Other protective mechanisms include earwax, urine flow, vaginal secretions, vomiting, and defecation. All of these help remove potential pathogens from the body. The body's first line defenses also include chemical factors that help protect against infection. For example, the sebaceous glands in the skin produce an acidic, oily film that helps prevent microbial growth. Perspiration, saliva, urine, vaginal secretions, they all contain lysozyme. This is an enzyme that can break down bacterial cell walls, in particular peptidoglycan. Gastric juice in the stomach contains hydrochloric acid. And as you can see in this image here, gastric acid, hydrochloric acid in the gastric juice makes it incredibly acidic with a pH of around 1. And this helps to destroy harmful microbes in the food and the water that we consume. Additionally, vaginal secretions, earwax, and urine, they're all mildly acidic. And this creates an environment that discourages the growth of harmful bacteria. The normal microbiota are beneficial microbes that live on the outside surfaces of our body, and that can be on our skin or within our respiratory system and gastrointestinal tract, and they help keep harmful pathogens out. How do they do this? Well, for one, they're simply just their presence there competitively excludes other pathogens from being able to grow. They're competing for a certain amount of real estate. And they also, have created ways to compete with other microbes by producing compounds that inhibit their growth. They also help to promote and engage and develop a healthy immune system. One example of their importance is in the vaginal microbiota. Broad spectrum antibiotics can kill these protective bacteria. And it can be often the case when someone is getting a course of antibiotics that this occurs, and this allows a yeast, which is not sensitive to those bacterial antibacterial antibiotics, And that yeast that is normally present in certain amounts, called Candida albicans, it is then allowed to overgrow and cause an infection. We discussed how this works with superinfections, and it's the same sort of mechanism there. Another example is the use of the antibiotic clindamycin. And this can disrupt a lot of different types of gut microbiota due to its broad spectrum activity as well. This increases the risk of infection by Clostridium difficile. Sometimes in the health industry, it's just referred to as C. diff. And this is a bacterium that can cause severe diarrhea and pseudomembranous colitis, which is a severe inflammation complication of the colon. These examples show how normal microbiota help maintain balance and prevent harmful infections. And this is why it's become such common medical practice to recommend that patients take probiotics while being on a course of antibiotics. Once a pathogen bypasses the first line defenses, the body activates its second line defenses to fight the infection. The formed elements in the blood play a crucial or important key role in this process. There are three major types that we need to be aware of. The erythrocytes, which are the red blood cells, they're responsible for carrying oxygen and disposing of CO2. The thrombocytes, which are also called platelets more commonly, and these help with blood clotting. And then we have leukocytes, and these include the white blood cells, sometimes just abbreviated WBCs, and these are responsible for fighting infections. Other important second line defenses include inflammation or generalized fever, which helps slow pathogen growth and recruit immune cells to the site of infection. And the body also produces antimicrobial substances that work to destroy or inhibit harmful microbes. These cells are generated from stem cells. Let me get out my laser pointer here, right here. So all of these cells are generated from stem cells in the bone marrow, and can fall into two categories: the granulocytes, which have visible granules in their cytoplasm that contain proteins and help break down bacteria and aid in phagocytosis. And then we have the agranulocytes, and these lack those visible granules. And if you look at the lineage here, you're going to find a plethora of cells that can be refined from the stem cells. Non-specific immune cells include the macrophages shown here, and the neutrophils shown here. So it can happen on both sides of these two major groups, and dendritic cells found here. And these three groups can respond to a wide range of pathogens. And just to explain here, platelets, which are here, these are small cell fragments, and they form an important part of our immune system. They serve to help with blood clotting. So those are the three main categories there. The three main categories of formed elements. We talked about the granular and the agranular leukocytes, and we're going to also focus in more on this chapter on the macrophages, dendritic cells, and neutrophils in particular. And let's just pause a little bit on these terms that we're using here. We use leukocytes, and we're going to use lymphocytes a little bit here. Leukocytes is a broader category. They encompass the white blood cells. These are cells of the immune system, and there's several types. There's the granulocytes, the monocytes, and the lymphocytes. So the lymphocytes, these are a specific type of leukocytes that are the B cells, T cells, and natural killer cells, or NK cells. And they all serve specific roles that we'll talk about for each one of them. All right, so let's go through those formed elements that are important to this class here. We have the neutrophils, and they leave the blood. They can enter tissues and destroy foreign microbes by engulfing them. This process of engulfing other cells is called phagocytosis. We've discussed a little bit about this when we went over the theory of endosymbiosis and how the idea was that there was a cell that phagocytized a bacterium, and that led eventually to the development of mitochondria or chloroplasts or a lot of other endosymbiotes that we find in nature today. The basophils in mast cells, they are capable of releasing a very important compound to the cell called histamine and other chemicals to trigger inflammation. And this is what can cause allergic responses. So you can imagine that these two types of cells are quite often involved in allergies that people experience. Eosinophils target parasites, and they can attach to their surface and release toxic proteins, including peroxides. We discussed how oxygen is very reactive, and some forms of oxygen are more reactive than others. Well, peroxides are very reactive chemicals, and that helps to kill parasites. So they can act on a very large scale, whereas what we've discussed so far is more about attacking microbes, very small cells, that is. The monocytes, they can leave the blood as well, and they can enter tissues, and they mature into the macrophages. These macrophages are able to engulf and digest foreign cells, again, phagocytize foreign cells in the body. Dendritic cells can also phagocytize microbes, and they activate the adaptive immune system by presenting antigens to T cells. So, I probably haven't defined this yet, but antigens are just chemical signatures of pathogens. So, an antigen is something that can be recognizable as a foreign pathogenic component of a invading cell, and they bring that to the T cell in order to help activate that T cell. NK cells, or natural killer cells, are capable of targeting virus infected and cancerous cells. So, virus infected cells and cancerous cells may exhibit something on their outside surfaces that allows for those natural killer cells to identify them. And those natural killer cells then release a chemical called perforin, which leads to the destruction of those bad cells. So, this is kind of unique from the other cells that we've talked so far, and that it actually targets our own cells, which have gone bad for one reason or another. T and B cells have an important role in the adaptive immune response. So we'll discuss more on this in chapter 18, but they can recognize specific pathogens and produce an immune memory. So they can recognize those pathogens after they've been identified as such in the future. And one last note here, the dendritic cells, the natural killer cells, and the T cells and B cells, these are all, again, examples of lymphocytes. One of the mechanisms of clearing pathogens is phagocytosis. And phagocytosis, it's the process by which a cell engulfs and destroys an invading microbe. This happens in several steps. First, we have chemotaxy. So the phagocyte is attracted to the invading microbe, and then it needs to attach to it. So adherence, the phagocyte attaches to the microbe. And next we have ingestion. The phagocyte extends the pseudopod, as you can see in this image here. See, it's a pseudopod being extended in this corner and grabbing that bacterium. So there's the bacterium, the Diplococcus. The pseudopod reaches out to attach to that and then consumes it. And so that brings us to the next steps. So step 4 is the phagosome formation. So this is a special vesicle, and it's called the phagosome. And this forms around the microbe inside of the cell. And step 5 the phagolysosome formation and microbe destruction. So the phagosome fuses with a lysosome, which we discussed in chapter 3, the cell, which releases digestive enzymes to break down and destroy that microbe within that phagolysosome. This process is a key defense mechanism, and it helps to remove those harmful invaders that have infected the body. And speaking of lymphocytes, we also need to understand what the lymphatic system is. The lymphatic system is different from our circulatory system. The lymphatic system is a network of vessels, tissues, and organs that circulates lymph fluid and plays a key role in immune defense. It's interesting because it has a unidirectional flow, meaning lymph moves in one direction, drains from these systems and towards the heart. It is linked to the circulatory system, helping to return fluids to the blood, and it moves lymphocytes throughout the body to detect and fight infections. B and T cells are activated in what we call the lymph nodes. These are specialized tissues around the body where they recognize and respond to pathogens in those areas and can be trained on pathogens in those areas. And this system helps coordinate the body's immune response and protect against disease. Something else we've talked about so far is inflammation. So, what is inflammation and how does it work? Inflammation is the body's local response to injury or infection. Local response. It causes pain, redness, swelling, heat in that area, and sometimes immobility at the affected site. The goal is to eliminate the threat of whatever occurred there and also begin the repair process through three main steps. So, a big reason why we have inflammation, as we recognize it with those different signs, is because of vasodilation. There are elements in the blood vessels that cause the pores in the blood vessels to widen and to open up, and that allows increased blood flow to the area. It also allows for migration and action of our phagocytes. So the white blood cells can then move into the blood and into the tissues of that area to help engulf and destroy any harmful agents. And it also allows for repair. So that damaged tissue gets more nutrients and repair factors brought to the region. And this helps to rebuild the damaged area after the threat is removed. This is somewhat related to fever, but fever is not localized. It's a systemic response. And so it raises the body temperature, and that helps enhance immune defenses. Uh, Pathogenic microbes tend to grow worse at higher than body temperature, so above 37 degrees Celsius. And also, our body's recovery mechanisms work better and faster at a slightly higher temperature. So, this helps to speed up recovery. It makes the body less hospitable to harmful microbes while supporting the function of our immune cells. The body produces antimicrobial substances as well to help fight infections. These include the complement system proteins, which is a group of about 30, more than 30 proteins. They're made in the liver and they circulate in the body, and they help to enhance the action of white blood cells. These complement proteins help to trigger inflammation in certain areas, as well as fever, and recruit immune components of the immune system to a particular region. Um, we have the interferons, and these are proteins released by virus infected cells that signal phagocytes to attack. So, if a cell is infected with a virus, it wants to basically self detonate and recruit phagocytes to the region, and so those phagocytes will help in the destruction of the viruses as well as infected cells. We also have the iron binding proteins. These help transport and store iron, keeping it away from microbes that need it to grow. And we have antimicrobial peptides. These are short chains of amino acids. That's what peptides are. So they're not quite full proteins, they're just short chains of amino acids that attack microbes by disrupting their cell walls and membranes. And these defenses all work together to slow infections and support our immune system. You may have heard of AMPs, or antimicrobial peptides, which again are very short, not quite proteins, or they're protein like. We call those peptides. These are produced by tissues or immune cells and the normal microbiota as well. They help protect the body by targeting a wide range of microbes, including bacteria, fungi, and viruses. The main mode of action of AMPs is to disrupt pathogen membranes, and this makes it harder for microbes to survive. One group of AMPs, called defensins, is made by epithelial cells, which are on the surfaces of your body, and macrophages can do it as well as neutrophils. And these can be secreted or act also inside of host cells where they damage microbial plasma membranes. They do this by forming pores in those membranes. And that causes, like we've seen with certain antibiotics, that can cause leakage of the components within the cell of those microbes, and that can lead to death or dysfunction. Another group are the bacteriocins, and these are produced by our microbiota, bacteria, and they inhibit competing microbes. They help also to maintain a balance in microbial communities. The immune system detects infections by recognizing PAMPs, or PAMPs. So these aren't AMPs, these are PAMPs. These are pathogen-associated molecular patterns. Pathogen-associated molecular patterns. Monocytes, macrophages, and dendritic cells all identify general molecular structures that are essential for pathogens to survive. So these structures, they're not found in human cells, and that making them therefore clear signs that there's an infection going on. There's some sort of foreign invader in that area. So examples of PAMPs, you could probably think of some if you compare prokaryotic cells to eukaryotic cells. It could be peptidoglycan, which is found in the bacterial cell walls, flagellin, which is a protein that's found in bacterial flagella. So while eukaryotes and prokaryotes can have flagella, this is specific to bacterial flagella. Oh, and by the way, the only cells we have that have a flagella are in male gametes, in sperm, and those do not have flagellin in them. Lipopolysaccharides, or LPS, which is found on the outer membrane of gram-negative bacteria, and also certain genetic signatures, sequences that are found in viral DNA or RNA. And by recognizing these patterns, the immune cells can quickly respond to infections. And I just want to make sure we're clear here. This is non-specific. These are things that are found, while, yeah, peptidoglycan is a specific compound, it's not the training of our immune system to memorize a certain structure. So say with a flu virus, you might have hemagglutinin or neuraminidase that mutate and change over time so that they're not recognizable anymore. And the immune system has to get retrained on the new shapes of the HA and NA molecules. Peptidoglycan is just always peptidoglycan. Flagellin is always flagellin. Liposaccharides, polysaccharides are always LPS. And so these are things that are considered non-specific. They're just recognizable markers that aren't really changing per se. You don't have to retrain them on them. Phagocytes use the pattern recognition receptors to detect PAMPs on pathogens, pathogen recognition receptors. PAMPs are essential for pathogen structure and function, right? Meaning they can't easily mutate them to avoid detection by the PRRSs, or the pattern recognition receptors. And in the next slide, we're going to show some examples of PAMPs, including lipopeptides, flagellin protein, and peptidoglycan, along with the PRRs, which are represented as green fork-like structures. So here they are. This image illustrates the pattern-associated molecular patterns, or PAMPs, and they're different colors here, and they can be found within the cell as well. And the green fork-like structures in all of these cases represent the pattern recognition receptors, which detect those specific PAMPs. So, for example, here we have the lipopeptide, and it's found in bacterial cell membranes. It's recognized by the lipopeptide receptor on immune cells. So this helps to keep gram-positive, gram-negative bacteria in check. Here's the flagellin receptor. Here's a peptidoglycan receptor. I really wanted to focus on this one, which is unique in that it's on the inside here. Here we find that we have the nucleic acid inside the phagosome, and this includes viral RNA or DNA, which is different and unique to that of the host DNA. Okay, so we have certain PRRs that can recognize that nucleic acid with our nucleic acid receptor. It triggers the production of interferons and other cytokines that help to fight those viral infections. And each PAMP is essential for pathogen survival. So these are pretty reliable, consistent products that can be recognized. Now, I brought up cytokines here on this previous slide that are something that can be secreted in interferons. Let's dig into this a little bit more. So cytokines, they are proteins that allow cells to communicate and coordinate immune responses. They help regulate cell proliferation, so that's growth, cell differentiation. This is where cells convert into specific cell types, and they help with inhibition of cell division. Obviously, if a cell is infected, you want to make sure it's not going through cellular division and multiplying that infection or helping in that process. And an important technical term that you need to memorize, this terminology, apoptosis. Apoptosis is when you basically pop a cell. It's programmed cell death. And next is chemotaxis. This helps cells move towards a signal, so you can bring in some phagocytes to that region. Some cytokines self-stimulate the same cell that secretes them. So they release a cytokine, and that causes a chemical cascade within that cell that released it. Interesting. While others travel short distances to nearby cells to send that message. And some even circulate throughout the body or through the bloodstream to affect distant cells. This image explains the three main ways cytokines function as molecular messengers in the immune system. These terms you may have heard before, the autocrine, the paracrine, and endocrine, describe actually how cytokines travel and act on target cells. Breaking down the terms, crine comes from the Greek word for secrete, so meaning cytokines are signals that are secreted and affect cells. Auto means self. We've learned that in metabolism when we talk about autotrophs. So auto means self. So autocrine signaling is where the same cell that secretes the cytokines, those interact with the cell receptors, and it is received there at that exact same cell and affects its behavior. Paracrine means near or alongside. And so paracrine signaling is when the cytokine is secreted to a nearby cell, which responds. And then lastly, we have endocrine. Endo means within. Endocrine signaling, where the cytokine travels through the bloodstream and reaches some distant cell within the body. And in this lecture, we're going to cover the three very important cytokines: interleukins, chemokines, and interferons. Interleukins are secreted by many types of cells, not just white blood cells or WBCs, and they regulate various functions, including immune system defenses. They promote the development and differentiation of T cells and T cells and B cells, or T and B lymphocytes, and hematopoietic cells. These are blood cell components. The chemokines recruit white blood cells, the leukocytes, to infection sites, and they help direct immune cells where they are needed. Interferons provide a non-specific defense against viruses. They stop neighboring cells from making mRNA, destroy RNA, and slow or stop protein synthesis. And this results in slower viral replication, helping to contain the infection. So this image illustrates the antiviral function of interferons. As we know, viruses infect cells, and the cells then go through this hostile takeover where the virus redirects the cellular machinery to making virions. And when a cell is infected by a virus, it releases interferons, shown here as these small red dots, to signal nearby cells and activate immune defenses. Interferons trigger three important responses. One, the unaffected neighboring cells destroy their RNA and reduce protein synthesis. And this is to prevent viral replication within them if they are also exposed to a virus. Number two, the infected neighboring cells undergo apoptosis. Again, that's a programmed cell death by that cell. It leads to its own death. And this helps to stop the virus from spreading by, if the cell kills itself, then the virus isn't going to be able to do that hostile takeover. And number three, immune cells are activated to enhance the body's overall defense. And this helps limit viral replication and spread in the body. So in chapter 17, we discussed the first and second line of defenses to prevent pathogens from invading, those general responses after a successful invasion. And in chapter 18, we are going to discuss the specific responses after a successful invasion. The adaptive immunity can be defined based upon where and how they occur. Adaptive immunity is the body's specific defense system, fine-tuned to recognize particular pathogens and to retain a memory for future protection. It has two main parts. Humoral immunity kills pathogens before they enter and infect host cells. It occurs in extracellular fluids, such as the blood and the lymph. Extra means outside of cells, right? And it targets pathogens before they infect host cells. It's mediated by B cells and antibodies, which bind to and neutralize pathogens. That's humoral immunity. Cellular immunity kills pathogens as they are inside host cells. It acts on infected host cells that already contain pathogens. It's mediated by T cells, or the T lymphocytes, which help eliminate infected cells. These two branches, they work together and they provide a long-term protection against disease. Antibodies are proteins that recognize and bind to antigens. Antigens are specific proteins, sometimes polysaccharides, that are found on pathogens. Antibodies are found on the surface of B cells, where they help detect antigens, and secreted by plasma cells, allowing them to circulate freely and neutralize pathogens. This recognition process is key to the humoral immune response, helping fight infections. And antigens, again, these are molecules that trigger an immune response. They're typically a part of the invading pathogen, unless, of course, it's a misidentification, like as with an allergy, where your body mistakes something that's not a pathogen as triggering an immune response. Now, these can include the cell wall, capsule or viral coats, fimbriae or flagellum, bacterial toxins. However, not all antigens come from pathogens. Some can be non-pathogenic substances that stimulate an immune response, like with our allergies. Epitopes are the specific parts of an antigen that the antibody can recognize and bind to. And this allows for a targeted immune response once those epitopes are identified. Just as a side note, allergies and organ rejection are both types or examples of your body reacting to non-pathogenic antigens. Most human antibodies are bivalent, meaning they have two antigen binding sites, shown here at the tips of these branches. Okay, that is where the antibody is shaped a certain way to be able to identify a epitope of an antigen that has a certain shape to it. So we can already see how specific these antibodies therefore are. They are made up of two identical light chains and two identical heavy chains. Okay, so here's in pink we see this is the light chain of the protein, and shown in blue this is called the heavy chain. And the light chain is only part of the branch, whereas the blue goes all the way down into a region we'll discuss called the effector region. Light and heavy refers to the molecular weight of the protein that comprises it. The stem, which is here at the base of the antibody, is called the effector region, or called the constant region. So, or sometimes the FC region. Notice it doesn't change from antibody to antibody. The shape up here at the antigen binding site, that is what's changing. So this is sometimes called the constant region as well. And the class of an antibody is determined by this region. So different classes of antibodies are going to have different constant regions. And this influences its function and the immune response, where it comes from, where it goes, what percentage they are, how long it stays in the body, things like that, what it specializes in. The ends of the branches of the antibody, again, are called the antigen binding sites. These sites are where the action happens and are built to be able to recognize and bind to very specific epitopes of the antigen. The role of antibodies is going to be incredibly important to the immune system, and so you should be able to easily draw and label the components that I discussed here: the light chain, the heavy chain, the antigen bonding site, where the antigen would therefore bind, the epitope, and the effector region. In addition, and because this course is focused on medical microbiology, you're going to need to know a little bit about each one of these classes. Now, I have highlighted in the red boxes the important stuff that I want you to know about each one of them, okay? It's probably also important, I missed one here, but that IgG is transferred to the placenta while the others are not. But these are what we call the immunoglobulins. Immunoglobulin it's a class of protein. It's almost exclusively composed of antibodies. So for all intents and purposes, the terms are used pretty synonymously. The structural aspect of those antibodies, we call them immunoglobulins based upon how they shape. They're kind of globules of proteins. But functionally, they act as an antibody, so they attack foreign bodies. So this table has a lot of working parts, it's going to serve as a great study guide. So let's take a look at each group of immunoglobulins here. Let's start with the most common immunoglobulin or antibody circulating around in all of our bodies, and that's the IgG. It makes up 80% of all of the antibodies in your body, and it has naturally a half-life. So how long does it take for whichever antibody is being made for half of them to disappear? It has a half-life of 23 days. So it lingers for a very long time in the body, hangs out for a long time, and helps provide an extended immunoprotection. And it is transferred in the placenta. Now, the IgM is a weird one in that you see all the other ones here. We either have them occurring all by themselves, or as we'll discuss with IgA, they appear as a dimer here, but they're usually singles or maybe a pair. IgM makes a pentamer. They have this weird formation where they form these disulfide bonds in their effector region, and they form this pentamer structure. Now, the IgMs have a specific job. They're especially effective against microorganisms and agglutinating antigens. These are antigens that agglutinate or glue things together. And very importantly here, these are the first antibodies produced in response to an initial infection. So once an infection occurs, it's a new type of infection, hasn't been seen by the body before. Maybe it's a new type of flu that you haven't had exposure to. Their body's going to train on IgMs first that are specific to the epitopes of that pathogen. So IgM is kind of like the first responder. IgA is pretty cool. IgA has this, what we call the secretory component, and this helps IgA to be released or secreted into tears, saliva, mucus, intestines, milk, and it's also found in the blood and the lymph. But so this is the antibody that can be passed on to infants through the milk, and it provides protection in a lot of these mucosal regions in the lacrimal apparatus as well. And so this allows antibodies to be localized and protect these different surfaces. Very important, right? The rest of these don't really do that. Next is the IgD. And the IgD makes up a very small amount along with the IgE. And these are present on B cells. And these help to initiate an immune response, these IgDs. Lastly, we have the IgE, very low concentrations, and these are actually bound to mast cells and basophils throughout the body. So as you can imagine, I discussed mast cells and basophils having the ability to release histamine. So these are often involved in allergic reactions, which is not good. But they have a very important role in protecting against parasitic worms, so different parasites of the body. All right, so let's now take a look at how this humoral immunity works, how things get activated, how these antibodies get selected, and B cells get activated, and these antibodies get selected for and reproduced en masse to fight off the specific reaction. Now, your body makes a bunch of B cells with a bunch of antibodies that are attached to them, and they are constantly making new ones all the time. They're just prototypes. And heck, they don't know what pathogens are coming out with, as far as like mutations that occur. They have no idea what's going on in the outside world. So they're just creating a ton of these B cells with antibodies that may or may not find something that is an antigen, may not find an epitope. They may find an epitope. One of them wins the lottery, so to speak. And there's a foreign invader, and it has that antigen which matches its prototype antibody. So that's the first step in the activation of this B cell. So that free antigen, it makes contact with an antibody on the surface of the B cell. And then that antigen, therefore, since it binds to that antibody, the B cell, what it does next is it engulfs and gets digested by that B cell. So we see that being broken and entered into the body. Those fragments of digested antigens are then displayed by the B cell surface. So it spits them back out and displays them. And that displayed antigen fragment attracts a T helper cell. And that T helper cell, it helps. It helps activate. So it's going to release cytokines that cause the B cell to become activated. And that B cell then differentiates into a plasma cell. And those plasma cells, instead of just having the antibodies on the surface, this antibody and B cell won the lottery. This is how the cell knows it needs to make a bunch of these. And so it starts releasing these antibodies and producing them on scale. Whereas the B cell, they don't do that. They just keep them on their cell surface to aid in its activation. These activated B cells into plasma cells can also be activated and differentiated into memory B cells. And these memory B cells, they remain in the body long term and enable a faster and stronger immune response if that same antigen or that same pathogen is encountered again. And that's what we mean by the memory of the adaptive immune system. Again, the humoral immunity kills pathogens before they enter and infect host cells. It's mediated by B cell activity and their corresponding antibodies. Now we'll move on to cellular immunity, which kills pathogens that are inside host cells. Cellular immunity, unlike humoral immunity, which is mediated by B cells, it is mediated by T cell activity. T cells have an important role in the immune system and are classified based on clusters of differentiation, or CD markers. These markers are glycoproteins. Glyco meaning sugar, protein meaning protein. These sugar proteins help T cells bind to receptors and carry out specific functions. Well, let's go through those. So the T helper cell recognizes antigens presented on phagocytes, such as macrophages and dendritic cells. And once activated, they produce their cytokines, as we saw in the humoral immunity. So they can produce cytokines that stimulate and coordinate the immune response. T regulatory cells help prevent autoimmune reactions. So they remove overactive helper T cells. This prevents the immune system from attacking our normal microbiota or fetal cells during pregnancy, so on and so forth, which, as you can imagine, is important. I described how the B cells, they just create different types of antibodies, and they're floating around looking for an antigen. So this is how the cell, one of the ways that the cell is able to differentiate normal microbiota and the self from actual pathogens, the T regulatory cells, keeping everything regulated. The cytotoxic T lymphocytes, or CTLs, they recognize infected host cells and work to eliminate them. They release perforin, which is a protein that creates pores in the target cell membrane, and granzymes, which are proteases. So ACE, remember, means it's an enzyme. So these break up proteins. These trigger apoptosis, or programmed cell death. And through these different roles, T cells help regulate and protect the body from infections while preventing harmful immune reactions. So we can see that these are all helping to prevent pathogen infections of cells, or clear them from the body. And this is turning into quite an interesting field. In 2017 was the approval for the first gene therapy for leukemia, or cancer of leukocytes. These are chimeric antigen receptor T cells. They're called CAR T cells. You may have heard them in the news. Chimera means they're genetically engineered to have different parts from two different types of cells. These T cells have been genetically engineered to produce an artificial T cell receptor that recognizes those cancer cells of the body and is used, therefore, to recognize and destroy those cancer cells by training your own immune system on those cancer cells. And so, after they've genetically modified your own T cells, they put them back into your body and they do their thing and they clear the body of cancer. All right, so the graph right here shows how the response of your immunological memory works. And on the x-axis, we can see the time of days of an infection, and the infection clears up, and then you get maybe a second exposure that happens at some other point in your life. Here, I don't know what it is, day 40 or something like that. Not too important. And this is the level of antibodies in the serum. And typically, we can see that after an initial exposure to an antigen, what's really important here is this second response. So I said IgM is typically the first primary responder. That's still true here. But you get an initial exposure, and some B cell wins a lottery, and it gets activated, converted into a plasma cell. You release those antibodies, and some of them are memory B cells, and they hang out in the body for a longer period of time. And if you ever get a second exposure, you don't have to go through that activation process and the B cell winning the lottery process, so to speak. You already have them ready to go and respond instantly upon a second response. So here we can see that the IgG just goes through the roof and results in a much more rapid and strong response. So instead of taking many days, you'll notice when you first get sick with a flu or a cold, it takes many days before you start to feel better. Well, here it's just triggered. I mean, you get a peak after just a couple of days in the secondary response versus here it's showing that's 7 days after the primary response here, 7 to 14 days. Okay, let's take a step back now and let's look at our adaptive immunity and how it can be categorized conceptually. Adaptive immunity can be put into two categories today. It can be described as being naturally acquired and artificially acquired. Naturally acquired adaptive immunity is ones that you get through some sort of incidental exposure to an antigen. So you're just out and about living your life and you get exposed to disease-causing pathogens, and you naturally develop your own adaptive immune response to that. And artificially acquired is adaptive immunity where you get deliberate exposure to the antigen. It comes from direct guidance from a medical professional. We can also break both of these categories actually into active or passive immunity. So active immunity is where the antigen enters your body naturally, of course, because we're looking at naturally acquired here, and your body produces its own antibodies. So it goes through that B cell activation phase, produces its own antibodies and specialized lymphocytes here. And so you actively produce your own memory to that particular pathogen and attack for that particular pathogen. Now, passive is when you don't do it. Naturally acquired passive immunity is where antibodies are passed from mother to fetus. And so we see in this example, you're making your own specialized lymphocytes and own antibodies, but in this situation, the mother is passing them to the fetus through either the placenta or via the mother's milk. And artificially acquired, we can break it down to the active or passive as well. Active, again, you have direct exposure to those antigens yourself. So this could be through a vaccine. The vaccine is injected into you. You develop your own antibodies and specialized lymphocytes. There's one other way that we can have a passive form through artificially acquired, And that's where instead of you being directly exposed to the antigen, you're just giving the antibodies. This occurs sometimes with anti-sera. So let's say you get a snake bite, and you're not producing your own active antigens through your own active antibodies here. You're going to be injected by, usually it's like a large mammal like a horse that's developed those antibodies already through special methods. It's been isolated and produced into an anti-sera, and you're injected with just the antibodies. And so that would be artificially acquired and passive, where you're not exposed to the antigens to acquire your own antibodies. There is a very long history of the development of vaccines. It's not something new. There's something that's been around for a very long time called variolation. And this was an early method of immunization against smallpox, in which a patient was inoculated with an attenuated or inactive smallpox virus. We'll talk about how that works a little bit. But this practice has been around since about the 15th to 17th century in China, later spread to the Middle East, Europe, and Africa. And the procedure involves using smallpox scabs. Those scabs are usually dried out, maybe exposed to sunlight, so UV light can hit they didn't know about viruses at the time, but the UV light in the sun, as you can imagine, is going to damage the nucleic acids in the virion particles, and that makes them less effective at causing an infection. And we call that attenuation. Attenuation means weakened. And those then could be dried out or whatever, and then they're exposed to the patient either through nasal insufflation, which is shown here in this image right here, and this image right here, by inhaling those dry scabs, or by placing the scabs into a small cut on the skin. And though effective in many cases, variolation, however, carried risks, as you can imagine. This could cause severe infection, and it may not always result in an attenuated virus exposure. And so this was eventually replaced by safer vaccination methods as we learned about the germ theory of disease. The development of vaccines began way back with Edward Jenner, modern vaccination practices with Edward Jenner, who was an English physician who observed that milkmaids who contracted a relative of smallpox called cowpox. They didn't get smallpox. And he hypothesized that infections with cowpox provided a protective effect against smallpox. This would mean that there was some sort of cross reaction with the adaptive immune system. To test this, he injected material from an active cowpox lesion into a young boy. The boy developed mild symptoms, but recovered and became immune to smallpox. Cowpox, while it could infect humans, it wasn't as virulent as smallpox was, so it was less harmful. While this would be a questionable ethical practice today, it was a little bit of a different world back then. And later, Louis Pasteur expanded on this idea. He developed vaccines for rabies, cholera, and anthrax. And this paved the way for our modern immunization. There are different components to a vaccine I just wanted to cover. One are adjuvants. These are substances that are purposely added to vaccines to help improve the immune response. So this makes the vaccine more effective by triggering a stronger immune response to the area. One of the most common adjuvants used in the past has been aluminum salts, and these have been used in vaccines for nearly 90 years. However, newer adjuvants are more lipid-based, so they help to stimulate the immune system in different ways by using different lipids that attract immune responses. Another type is called CpG. This is a synthetic nucleotide, And what it does is it mimics bacterial and viral DNA, and that triggers a stronger immune response as well. Live attenuated vaccines expose an individual to a weakened strain of a pathogen, and that allows the immune system to develop protection without causing a severe disease. These vaccines are really good at eliciting a good, strong response. They stimulate both cellular immunity, that's the T cells, and humoral immunity, the B cells, leading to a strong and lasting immune response. Because they use a live but not weakened pathogen, or an active one in the case of viruses, they do not require adjuvants, and often they need fewer boosters or none at all. However, they cannot be used to immunocompromised individuals because even a weakened pathogen could still cause illness. So we have to be careful here. There is a small risk that weakened strain might revert back or mutate back to a more virulent form. And an example of a attenuated one would be the oral typhoid vaccine, which is given in capsule form if you ever travel outside of the country. It's common or mandatory that you get the oral typhoid vaccine. Inactivated vaccines use a whole pathogen that's been killed or inactivated, so it cannot cause disease. Because this pathogen does not multiply in the body, it's less of an antigenic and less of a immunoresponse than an attenuated one, so it can require adjuvants and booster doses to maintain immunity. Inactivated viruses primarily stimulate the humoral immunity, or the B cells, which produces antibodies to protect against future infections. Examples of these include the influenza vaccine or flu shot. And no, the flu shot does not give you the flu because the virus is inactivated. Finally, we have subunit vaccines. These contain only the most antigenic parts of a pathogen rather than the whole organism. Because they use only the key components, multiple booster doses are required to maintain immunity. An example is the H. influenzae type B or Hib vaccine, which uses part of the Haemophilus influenzae's glycocalyx to stimulate an immune response. The Hib vaccine helps protect against serious infections like meningitis, pneumonia, and is recommended for infants as part of just their routine immunization. Toxoid vaccines protect against bacterial exotoxins by using an inactivated toxin to stimulate an immune response. These vaccines only stimulate humoral or immunity B cells, helping to produce antibodies against the toxin. Examples include the DTaP and Tdap vaccines. One is the initial, one is the booster, but these are that abbreviation is that it helps protect against a lot of infections from tetanus or T, diphtheria D, and pertussis or P. The AP means acellular pertussis antigen, so there's no cells of pertussis there. The Tdap is the booster that you often will get, especially if you're working in the health profession. Conjugate vaccines help the immune system recognize capsule polysaccharides, which would otherwise be poorly antigenic. Our immune system's really good at recognizing proteins, very poor at recognizing polysaccharides, our B cells, our antibodies, immunoglobulins. And this is done by binding the polysaccharide to a harmless protein. So that's why C-O-N means with. So it's bound with that harmless protein, but that makes it more detectable by the immune system so that it can bind to that antigen binding region. An example are Streptococcus pneumoniae vaccines, Pneumovax 23 and Prevnar 13. You don't have to memorize those, but these can protect against pneumococcal infections. And that wraps up our discussion of the innate and specific immune system. Hopefully you gained a lot of great information here. I think this is going to be a big focus of whatever most of you do in the future, as most of you are going into the health profession. So it was a little dense on material, but I hopefully you gleaned some good information from this one.